Okay. So, do you have questions before we start? So today we just only want to talk about how the neurons do the job. Right? How the neurons do the job. This is very important. There's only one topic today, which is the functions of neurons. Remember, there are two types of cells in nervous system, in nervous tissue. One is called neuron, which is the functional cell, the one that carry information, process information, reorder everything. And then the second one is neuroglia, which is the supporters for the neurons. Now, the question is how the neurons perform the functions. Okay? How the neurons send information, how the neurons carry the orders, how these cells actually communicate. Do the neurons have mouth? Do neurons have mouth? No. If neurons have mouth, it's easy, they can order. Do that, okay, but the neurons or your cell do not have mouth. How the cell communicate? Using this actually, okay? Cell communication, okay? Using chemicals. Okay, so chemicals are used for communication inside our body. Therefore, chemical balance is really important in our body. If our chemical is not balanced, let's say in your brain you don't have enough chemicals or neurotransmitter, you might be crazy. You have a mental problem. That's actually chemical imbalances. Okay? There are two chemicals that is really important for communication, cell-to-cell communication. The first one is called neurotransmitter. Okay, neurotransmitter. And this neurotransmitter produced by neurons. Right? So when the neurons order your skeletal muscle, they release the neurotransmitter. Okay? The second one is called hormones. The second chemical used for communication is called hormones. Hormone also will tell some organ to perform something. Okay? So this hormone produced by what you call the organ that produces hormone. It's called the endocrine gland. Produced by endocrine glands. Neurotransmitter usually produce a very fast action. The hormone slower actions. Okay. So when the neurotransmitter release and tell an organ to perform something, usually the action is very fast. But hormones, they perform slower action. Okay? Okay, now we're gonna go back to here, to number one over here, which is the neuron function. Okay, how neurons perform its functions. Okay, now before we go there, okay, we have to understand this first. I believe we already talked about this when we talk about cell and the cell membrane. Okay, in the cell we have many channels. We have many channels. Okay. The channel that used for sodium is called what? Sodium channel. Let's say this is sodium channel. Sodium channel. The cell that used for potassium is called potassium channel. That's like that. And remember, there are more potassium inside the cell. You still remember that? And there are more sodium outside the cell. You guys fine? Now, when the sodium channel open over here, what happened with the sodium? In or out? In. And you still remember how many sodium enter into the cell whenever they use the channel? How many? Good? Yes, 3. 3 sodium enter. Okay? And because the potassium channel open over here, what happened with the potassium? In or out? Out. How many potassium will be out? 2. You with me? Okay? And then what happened with the cell if happening like this? The cell gonna be continuously positive because there are extra 1 positive over here. Yeah? The cell will not do that because cell need equilibrium, homeostasis. Therefore, we have pump. You still remember the pump in the cell? What is the name of the pump? Sodium, potassium, ATP pump. Yeah? Or sodium potassium pump. They require ATP. Yeah? And this pump will pump how many sodium? 3. Remember, sodium enter 3, then it should be pump 3. Out or in? Out, because more sodium outside, that should be pumped, right? 3 sodium out, but it's gonna be make it balanced. But still, we have another one over there. What is that? Potassium. So potassium will be pumped in or out? In. How many? 2. Okay, it will balance this. This is homeostasis, and this is what we call the R or RMP, resting membrane potential. Okay, and when we measure, okay, all cells will have resting membrane potential around negative 73 microvolt. So inside the cell, the potential actually or the electricity actually more negative. What is the resting potential? Negative 70. You get this one first. Okay? Now, in order to make this cell doing action, then this potential has to be changed, whether increase or decrease. Are you with me? Okay? If the potential increase, it means that, let's say, a lot of more sodium enter into the cell. Okay, what happened with the cell is gonna be? Increase the potential. What is the number gonna be? Higher than negative 70 or lower than negative 70? If a lot of positive coming, higher. Higher, increase, positive. If it is reaching minus 55, at least minus 55, it means that how many positive actually enter to reach minus 55 from negative 70? Around 15. Is that's correct? Well, around 15 positive enter, it will change the potential into this, which is increase. Sometimes it can reach plus 35 microvolt. It means but around 100 maybe or 50 positive coming, right? Because it can increase a bit. But this is the what we call the threshold. Threshold. Threshold to change the action. So if or whenever the cell reach negative 55, then there will be action. And mostly it's called the excitability. Excited. In this case, if it is, for example, the muscle cell, it will be contracted because it's excited. Oh, contracted. Right? Let's change this resting into higher, positive. Okay, everything is positive, they're gonna be excited. Okay, now let's say the one that is open over here, let's say this chloride cell. We have a chloride channel. We have a lot of chloride outside the cell. Let's say the chloride channel open. What happened with the chloride? In or out? It's going to be in, right? Because this is diffusion. Let's say in. So what happened with the RMP if the one that enter is chloride? More negative or more positive? It's going to be more negative. Let's say reach negative 90, about 20 negative coming, right? So when it is happening, going to the negative, the cell will be inhibited. Okay? The cell will be inhibited. The cell will not be active. You follow this? Okay? This is how to make cell become active or inhibited by changing the voltage, by changing the potential. What is the resting potential? Negative 70. Okay? Can be increased or decreased based on the action that's coming. This is the basic for understanding how the neurons will work. Now we go to the neuron itself. We're gonna look what happened with the neuron. Okay. Now, ion channel. Okay. We're gonna learn about the ion channel first. There are two types. The first one is called the leakage. Or keep opening. Okay. The second one, there is a channel that actually have gate. Okay, it's called a gated channel. Okay, there are three types of gated channel. Okay, I'm gonna put here three types of gated channel. The first one, I'm gonna use mechanical first. Mechanical gated channel. Okay. What is this? This is the ion channel that may be open or closed based on mechanical stimulus. Okay, open or closed due to mechanical stimulus, such as touch, vibration, for example. That's mechanical, right? Follow this? The second one is called voltage. Voltage gated channel. This channel open or close due to voltage. Increase or decrease of voltage. It might open or close the voltage gated channel. And the last one is called the ligand gated channel. Okay, this one open or close due to chemical. If there is chemical go to this channel, attach to this channel, then what happened? The gate open or close, depend on the type of chemical. But it is based on the chemical, ligand or ligand gated channel. You follow this one first, right? There are three gated channel, mechanical gated, voltage gated, and the ligand gated channel. Okay, I'm gonna erase this. I need place for drawing the the neuron. Okay, are you finished? Not yet? Okay, I'm gonna wait. Done? Okay. Now, okay, so in the cell, we might have linkage channel that keep open or gated channel that open or close based on stimulus, type of stimulus. Let's say this is a neuron. Okay. Okay. Let's say this is a neuron. What you call the first part of the neuron over here? Dendrite. Good job. Dendrite. Eh? There are always many of them. Eh? And then what's the second part?
Cell body.
Yes. Cell body. Alright? What is the third part?
Axon.
This is the axon. Okay, now remember there is gated, there is channel over here. Channel. Okay. There are some channel that linkage open and there are some the one channel that is gated. We will not talk about the one that is keep open because that's actually the one that for maintaining the homeostasis. The one that is really important for changing the potential is the gated channel. Okay, because this is the one that can be controlled, whether open or closed. You get this one? Okay. Now what is the RMP? What is the RMP?
Resting membrane
Yeah.
potential.
How much?
Negative.
Negative 70. Okay, negative 70 microvolt. When the cell, when the neuron has this potential, negative 70 microvolt, what happened with the neuron? Resting, okay, not doing anything. It's resting, right? It's not giving any action or order. Okay? Now, let's say somebody touch us. When somebody touch, then there will be received. Let's say stimulus over here. And the stimulus is touch. Okay? Now, let's say the stimulus will go to where? Which one does receive the stimulus? The?
The
The dendrite. Eh? The dendrite. When the dendrite receive it, what happens? It will cause the gated channel over here open. So there is sodium gated channel here on the cell body. Right? It's gonna send that stimulus, and then what happens? Sodium gated channel open. What happened with the sodium? In or out? It's gonna be in. Okay, in. It's depend on the frequency and also the power of the stimulus. The more power, the touch, for example, or pressure, the more gated channel will be open. Then the more sodium will be entering into the cell body. You with me? Now, because of this, because of this information, what do you call this gated channel over here? The sodium gated channel on the cell body. What the type gonna be? What type of gated channel will be? No, it's open because of touch. Somebody touch you. There are three types of gated channel. Read it again. Okay, yeah. There are three types of gated channel, right? Mechanical, voltage, and ligand. You with me? Now, because somebody touch you, what is touch? Mechanical. It's open the sodium gated channel. Now, what type of gated channel this is gonna be? Mechanical. Sodium, mechanical gated channel. Right? So in the exam, I might asking this question. If it is open because of some stimulus, what is the type of channel it's gonna be? Okay, for example, this one. So the gated channel, the sodium gated channel in the cell body will be mechanical gated channel if it is open due to the mechanical stimulus. Okay. Now when it is enter, let's say this one enter only 10. Let's say 10 sodium enter. What is the change gonna be? From negative 70 into negative 60. What happened? Yeah. Is there any action over here? Remember the threshold. What is the threshold? What is the threshold for action? Negative 55. This one higher or lower than negative 55? Lower. It means that nothing happening. Okay, so nothing happening. Therefore, okay, for the neuron action, there is a rule called the all or none. There is a rule called the all or none. What is all mean? Whenever it's reached negative 55, then the action will be travel all the way until go to the next cell or go to the effector. It will not stop. Okay? But if the threshold is not reached, what happened? None. Nothing happened. So the cell just like a resting time. Nothing happening. Are you with me? So this is the rule, all or none for the neuron action. Okay, so the threshold should be negative 55. Okay, now let's say because the touch had more stimulus coming, then what happened? More sodium channel open, and finally it's reached, let's say plus 10 microvolt. Okay. And so this is gated channel open. And finally it's reached this one. Okay. So what happened this? There will be action or not? Yes, because follow the rule over here now. Now what happens when it is increased at least negative 55, it actually will make or stimulate the opening of sodium channel over here, sodium gated channel in the action. Are you with me? When it is reached the threshold in the cell body, it will stimulate the opening of sodium gated channel. Now what do you call the sodium gated channel over here? Open because of this. Yes, we're gonna be voltage gated channel. Can you follow this? So voltage gated channel is gonna be voltage gated channel. So this is enter, sodium enter, plus enter. Finally, the plus gonna be actually go or travel from cell body to the end terminal. Okay, you guys right? So it's going to increase. Let's say this is also positive 10 microvolt. Are you with me over here? Now in the end terminal, the axon terminal. So this is what we call the axon terminal. Okay. There is another type of gated channel which is calcium. The calcium gated channel over here. Okay, calcium gated channel. And the same with the sodium, we have a lot of calcium outside our cell. So what happened with the sodium gated channel open? What happened with the calcium gated channel open? Calcium will enter. Now remember, this calcium gated channel open because of this, because of the positive charge that travel from the cell body to this N-terminal. Now what do you call this gated channel over here? Calcium gated channel on the N-terminal. It's open because of voltage over here. It's going to be voltage gated channel. So calcium voltage gated channel. You follow this? And remember last time when we talked about the motor neuron that go to the, so let's say this is muscle cell. Okay? So in the N-terminal there is a vesicle, okay, that carry neurotransmitter, right? Neurotransmitter. Okay. If it is muscle cell, do you still remember what is the name of neurotransmitter that go to the muscle cell? No, the name of neurotransmitter, not the neuron, not the name of the neuron. The name of neurotransmitter that make our muscle cell contract. Yes, ACH. Remember that, right? ACH. So this neurotransmitter, the one that go to the muscle cell, is called the acetylcholine or ACH. You follow this one, right? Now what happens when the calcium enter into this axon terminal? It will push this vesicle into the membrane, and then it's going to be released. The neurotransmitter, in this case again, this acetylcholine, to the area over here. What do you call this area? It's called the synapse. Remember that? Synapse is the area between neuron to the next cell. And because this synapse used by chemicals, this synapse is called the chemical synapse. So synapse that used by the chemical, and acetylcholine is chemical, right? It is chemical. Right. Now, what happened next? Let's say this is muscle cell. We're gonna kind of review what happened with the muscle cell. In the muscle cell, remember, we also have the sodium channel, right? That will be open or closed because of the acetylcholine. Right? So when the acetylcholine, let's say acetylcholine attached to this channel, sodium gated channel. Right? It will open the sodium gated channel, and then what happens with the sodium will enter and go to the T tubule. Remember, go to the T tubule. Go to the inside the cytoplasm of muscle cell. What do you call the cytoplasm of muscle cell? Sarcoplasm, right? Sarcoplasm. Now we go here first. So this is sodium gated channel that is open or closed because of the acetylcholine. Now what do you call this gated channel? Open and closed because of chemical. That's the ligand gated channel. You follow this? This is the ligand. This is example of ligand gated channel. Okay. So that's the story how this neuron actually works by opening the gated channel, change the voltage, and then at the end over here, it will release the neurotransmitter. Neurotransmitter is chemical, will be attached to gated channel in the effector cell. In this case, it's muscle cell. Open the channel. So this channel is gonna be sodium ligand gated channel. And continue the story. What happened? The sodium, which is positive charge, will enter into the sarcoplasma, and in sarcoplasma we have reticulum that store the calcium. What do you call that? Yeah, the SR, right? We have SR that have a lot of calcium in it. Yeah, remember that? And the SR actually have gated channel also on the membrane. So this is calcium gated channel, right? And that gated channel is open because of the voltage over here carried by the sodium ion. Now, what do you call the gated channel in the SR? What type is gonna be? Voltage. Gonna be calcium voltage gated channel. Okay, now continue the story. What happened with the calcium? It will out from the SR, where the calcium will bind to which protein? In the sarcomere, troponin. When it binds to the troponin, open the tropomyosin on the binding site for the head of myosin to bind, and then finally pull the muscle contracted, the sarcomere. And this is, okay, when it is increasing the voltage over here, this is always excitation. This is what we call the APSP. Okay? ASP, okay, excitatory post-synaptic potential. Okay? So APSP means the cell is become positive, become excited. That's APSP. In this case, when the muscle cell receive the acetylcholine, it will be contracted. But if, for example, the one that coming, let's say there is a neurotransmitter called GABA, okay? So GABA is another type of neurotransmitter. When the GABA release into the cell, let's say this is a cell, okay? This GABA is actually open the chloride gated channel. Okay, so this GABA will attach to the chloride gated channel and causing the channel to be open, and the chloride will enter into the cell. Now, what happened with the cell? The potential inside the cell will be more negative or less, more positive? More negative. When it is more negative, what I mentioned before, it will be inhibited. Okay, so if the cell become negative, okay, the action or the product is called the IPSP. Okay, inhibitory post-synaptic potential. Okay, so when the cell become negative, it's become inhibited. Now, what type of gated channel over here in this cell? If the chloride channel open because of GABA, what type of gated channel is gonna be? Ligand, because GABA is also chemical, right? So this is gonna be ligand gated channel. Examples, okay? So GABA is actually produced by our neurons in our brain. It will actually affect other cells in the brain, causing that cell become what? Inhibited, become relaxed. Okay, there is another chemical that act like GABA. Have you ever heard this chemical? Valium. What is Valium used for? Anybody use Valium over here? It's a drug. Drug to make you fly. Why it is fly? Because basically your brain is inhibited. Your brain doesn't work. You doesn't want to do thing. You just want to, I don't know, sleep or whatever. Okay, that's the example of chemical that can actually affecting your GABA over here. It's kind of replacing the GABA. Now, what happened with people that use Valium over and over?
Their brain is rot.
Right? And also, their GABA actually will not be produced. What happened with them if they need it to be relaxed? They need more Valium, right? So they become, what do you call it? Addicted to the drug. That's actually the drugs, what the drug use or do to our brain is change the normal neurotransmitter. There is another neurotransmitter, it's called the endorphin, that also IPSP in the brain. What does IPSP mean? Inhibit brain function. It is needed for us. Like for example, we have pain, for example, the endorphin will be produced, and our brain will try to forget the pain. Are you with me? And we produce this naturally. There is another drugs that people take it, is called this. You see the name is very similar. Morphine, because the chemical is really similar in structure. So this morphine actually will causing the endorphin will not be produced in our body if we use it over and over. You follow this? So morphine is actually kind of agonist of the endorphin. It will replace the endorphin. It will become, addicted to this. Okay? So this is examples of action of neurotransmitter. Maybe there are some neurotransmitter that APSP, and there are neurotransmitter that IPSP. And you can read that on the, I think we have it in this PowerPoint. This is the same, this is just the process, similar. I just use it as one picture over there, what's going on, which is open and etc. Okay, this is the resting potential. If potential increase, it will increase the potential at least negative 55. Okay, and this is what we call the depolarization. Whenever the potential increase is called the depolarization. Whenever the potential decrease, like in APSP, is called the hyperpolarization. Okay, so increased potential is depolarization. Okay, so this is also in the action. There's a stimulus. Whenever it reaches negative 55, which is the threshold, so this is the resting point, right? It will increase. This is what we call depolarization. What happened during depolarization? The sodium channel, gated channel open. So a lot of positive coming. And then repolarization, what happened? This potassium channel actually that will be open. Because potassium, when it is open, the gated channel open, what happened with the potassium? Will out from the cell. The cell become negative. Okay? Repolarization and going back to the resting potential. Sometimes the potassium channel keeps opening, the sodium channel is not open yet. Then what happened? It's actually overshoot, going down to the usually negative 90. This is what we call the after hyperpolarization. Okay, so this is, and then finally it will go to the resting time, and then going back again, continue like that. Okay, so that's potential changings in the neurons, especially in that action. And again, this is what happened during that action potential. Basically, sodium gated channel and potassium gated channel will be do the job. This is just example how the drug actually changing the action in our neuron. Like for example, novocaine, lidocaine, what is used for? Anesthesia, right? What does anesthesia do? Actually, is blocking the sodium gated channel. It will not open. If it is not open, what happens? It means that the cell will be, or the neuron will be interested. It will not send you any information. If it is like put it here and then it's really pain, but because the sensory neuron is actually blocked, the sodium channel is blocked, what happens? The sensory neuron will not carry that information to your brain, and your brain will not know that this is actually hurt, okay? Because it's blocked by this drug, by this anesthesia. And we already mentioned this, there are two types of conduction, which is saltatory. This is for the neurons that do not have the myelin sheath. What type of neuron is that? Type
C.
A, B, or C?
C.
Type C. Right? And saltatory is for the neuron that have myelin sheath, gonna be
A.
faster, A or B. Right? So A is the fastest one. Okay, this is how it works. So again, this is continuous conduction, boom, 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 just walking. But this one, boom, boom, boom. Because the sodium channel for the neuron that have myelin sheath is only on this area over here. It's called the node of Renfier. This is where the sodium gated channel is located. So therefore, it's going to be open here, open again here, and then finally will kind of giving us the saltatory or the jumping action potential. I already talked about this one. There are two types of synapse actually, electrical and chemical. The one that we have over there is chemical synapse. An example of electrical synapse is between the cells in your cardiac muscle, because your cardiac muscle has a gap junction. So the sodium ion is just go from one cell to the cell using that gap junction, using that hole. It doesn't have to release the neurotransmitter over here, okay? Because the cell has the gap junction connection using that hole. Yeah, that's actually the same picture, but this one is better. So, this is a chemical synapse, neurotransmitter release into the synapse, attached to the channel. It's gonna be gated channel over here, because it's open by the ligand, the neurotransmitter. This one is called the ligand gated channel, whether sodium, potassium, or chloride, any channel. Whenever it is open because of chemical, then the name gonna be ligand gated channel. Okay, that's similar with that one. So the release of neurotransmitter. Yeah, I already talked about this one. Yeah, the calcium channel open push this neurotransmitter. This is on the N terminal. Okay, this is happening in the N terminal. The calcium channel open because of this voltage. So this calcium channel will be voltage gated calcium channel. Go there and then open it and then doing the action on the effector cell. Okay, again there are two types of effects on the effector, maybe EPSP or IPSP, and I already mentioned that. If it is EPSP, it means that excited, there will be action, but if it is IPSP, it's actually inhibited, there will be no action. Okay, so stop the action. After the use, the neurotransmitter have to be removed. Otherwise, if it is not removed, it will keep giving the orders. Okay, there are three types of removal: diffusion into the N terminal. So basically, this acetylcholine can go back to the vesicle, or erase it, or remove it by the enzyme. It's called the acetylcholine esterase. Or there is a glial cell, okay, microglia or neuroglia, that can also remove the neurotransmitter from the synapse. So this is the three ways to remove it. Okay. Again, the threshold is negative 55. Anytime it is below that, there will be no action. Yeah. It can be increased by 2 neurons or just 1 neuron with 2 or 3 axon terminals. So it can increase the potential on the area to reach at least negative 55 over here. So before, let's say here, before it's reaching negative 55, there will be no action. But when you're adding plus 1 from here, 1 from here, and then finally it will reach negative 55. Whenever it is reaching negative 55, it's going to be all action. Remember the rule, all or none. So it's going to be going back here. Okay, these are example of neurotransmitter. There are more than 100, and this is only several of them. And we already mentioned about acetylcholine. This is APSP in the muscle cell. Exciting. Right? We already mentioned about GABA. What is GABA? IPSP. This is inhibitory. This is excitatory. This is inhibitory. This is another. This one, okay, can be inhibitory, can be excitatory, depend on the location. Okay, so this is just explanations, the function of each neurotransmitter. We already mentioned about acetylcholine. This is APSP in the muscle cell. Glutamate is also APSP. GABA is IPSP, inhibitory in our brain. Dopamine also regulating skeletal muscle tone. Okay, so like people that have Parkinson's disease, they actually do not produce dopamine. So what happened with their muscle tone? They cannot control it. They gonna keep shaking. Okay, because this dopamine is also used for controlling skeletal muscle tone. And this is IPSP, the enkephalin, endorphin, the norepinephrine. This is actually pain reliever in our brain. This is natural pain reliever. And there are some drugs can be used for as an agonist or antagonist. Agonist means increase the reaction. Agonist means can opposite of the action of the neurotransmitter or hormone. So neuron have circuits. So this is just an example of neuronal circuits in our brain. And about repair, we already mentioned that nervous tissue, nervous system, they usually cannot be repaired. Okay? So only several parts. Okay, so maybe PNS, they have still a little bit ability to be repaired, only the action part, not the cell body or dendrite. I think we're done. Okay? So your assignment will be due next week, right? So assignment due next week. And for the one that have left today, don't forget, make sure you watch the video first, because I will not explain again in the lab for the special senses. We don't have time because there's a lot of activities during the lab today.
The video you were back on the last video.
No, that's not for the lab quiz. The lab quiz is for previous, for only for the brain, not for the special senses.
So it's just the last one, the one we wasn't there.
Yeah, the one you wanted to right.